In this video, we're going to be talking about the factors that influence enzymatic reactions. And so there are multiple factors that will determine how fast an enzymatic reaction will occur. So the amount of the substrate, and so think about if you have a lot of substrate, then a reaction will occur faster than if you have very little substrate, because enzymes and substrates, they don't see each other, but you're going to have more effective collisions if you have more molecules in an environment. Temperature. So if you are increasing temperature, then molecules move faster. And decreasing in temperature will cause molecules to move slower. pH. So the acidity or the alkalinity of the environment can influence enzymatic reactions. All enzymes have a specific pH that they work best at. So outside of the optimal pH will cause them to decrease in activity. Okay. Enzyme inhibitors. So anything that is going to inhibit enzymatic reactions. Oh, hello, no. So if something causes an enzyme to not be able to function properly. Activation of enzymes. So some enzymes are inactive. Whoa, okay, let me write that again because that looks really bad. Okay. Tion, there we go, that looks much better. Of enzymes. Some enzymes are inactive normally, and so they only become activated when they are needed. And that's really actually important. And then cofactors. So cofactors are molecules that help enzymes to do their job. And so we're going to look at each of these a little bit more closely. So when we talk about substrate concentration, enzyme activity will always increase as substrate increases. And so the more collisions that enzymes and substrates can have, the more products that will be produced. And so I'm just going to draw this. So say this is an enzyme and this is the substrate. Okay, so if you only have one substrate, right, then only one reaction can occur. And say this enzyme catalyzes 10 reactions every second, okay? So it works 10 times per second. If you only have one substrate, you're only going to get one product. Make sense? So the more substrates you have, the more product you can have at any given time. So it's going to increase activity. If you have, 10 substrates, 1, 2, 3, 4, 5, 6, 7, 8. I realize my substrates are not very neat, but whatever. Then at the end of 1 second, you could catalyze all of these reactions potentially, right? But if you have more, then you're more likely to have these enzymes and the substrates attaching and the product being produced. So enzymes work most effectively when there are more substrates. And actually when all enzymes are working at their optimum level, then they're actually considered saturated. So that's called enzyme saturation. And that means the enzyme can't work any faster because it's already doing its optimum level. So in this case, every second, this enzyme would be catalyzing 10 reactions, producing 10 products from the reactants. Okay. So temperature can influence enzymatic activity. All organisms have, all enzymes have specific temperatures they work most effectively at. And so if the enzyme works in a cold environment, they're going to work better in that cold environment. And if you heat it up too much, that can actually affect the activity of the enzyme. If you increase, or if they work in a warm temperature and you cool it down, the same thing can happen. So heating something up too high, because enzymes are proteins will denature at high temperatures. And so what that means is their shape changes. Loss of shape. And because the structure determines the function, that means they also loss of function. They lose their function. So high temperatures can actually cause protein denaturation, which means they've lost their shape and function. They can no longer work. If you cool the environment down too much, these proteins will actually become more rigid. So remember, enzymes have that induced fit, so they're not very rigid. They have to be able to hug their substrate a little bit. And so when they become more rigid in cold temperatures, they don't work as effectively, so they slow down. And it's kind of like your own body. When your body is cold, you can't work as effectively. And if your body heats up too much, you can actually lose cells. Think of that. So in endothermic organisms, and so that's what we are. We're warm-blooded or endothermic. The temperature promotes reaction rates, whereas in ectothermic organisms, so cold-blooded organisms here, the temperature limits the reaction. Okay. pH also can cause denaturation to occur. So in pH, though, enzymes all work at an optimal pH. So the pH of an enzyme could be around neutral. So remember, 7 is neutral. Or they can be very acidic. So anywhere down here is acidic. And then anywhere up here is alkaline. And so enzymes are going to have their optimum pH. If we move an enzyme, and I'm just going to use pepsin as an example, if we change pepsin's environment, make it more alkaline, so we move it from this acidic environment and move it to an alkaline environment, it will cause denaturation of that protein. If we take trypsin, who works best at more of a neutral pH, between 7 and 8, it works really good and starts to stop functioning at around 9. If we increase the alkalinity or decrease the pH and make it more acidic, it will also denature. So the pH is very important. We have to maintain pH of every enzyme for its function. Enzyme activation. So we have some enzymes that don't function until they're needed. So I teach anatomy and physiology, so I use humans as an example a lot of times. And one example, which you just saw in the last slide, is pepsin. Pepsin is a protein that breaks down protein. So it is a protein enzyme. It's a protein that functions in breaking down meat. And it's found in our stomach. It is produced by cells in our stomach. And these cells secrete pepsin as an inactive enzyme called pepsinogen. So the pepsinogen then comes in contact with hydrochloric acid, HCl, which is found in our stomach acid, or in our stomach as part of the gastric juice. And the hydrochloric acid actually cleaves a portion of the pepsinogen, producing the active form, which is pepsin. Okay, so that's one example. Another example, we have proteins that are floating in our blood plasma all the time called complement proteins. Complement proteins are inactive normally, but they can become activated if they are cleaved or if they come in contact with bacteria or any pathogen. Enzyme inhibition. So inhibitors can also cause enzymes to not function properly. There are two major types of inhibition. So one is written as the plugging of an active site. So I call this competitive inhibition. And basically, you have some imposter, some other molecule binding to the active site and causing the enzyme to not be able to bind to its normal substrate. The other is called non-competitive or allosteric inhibition. So allosteric inhibition occurs when a molecule binds to an area outside of that active site. Actually, that spot is known as the allosteric region, which is where the name comes from. And when it binds, it changes the shape of the active site. And so both of these are actually used in feedback regulation to control metabolic pathways. So if we don't need something to be produced, we can actually stop them from being produced. But competitive inhibition also can occur when you have poisons in your system. So let's look at that, okay? So here's what competitive inhibition looks like. Here we have an inhibitor. So this molecule can bind to the active site just like its normal substrate does. But when it binds, it doesn't unbind. And so now the substrate can no longer bind effectively. And then in non-competitive or allosteric inhibition, we have this region here known as the allosteric site. And so a molecule binding to the allosteric region changes the shape of the active site. So now the substrate can no longer bind in the active site because it has been changed. So I did mention that both of those are used in maintaining enzymatic pathways, metabolic pathways, and in inhibiting reactions from occurring that don't need to occur. And that's known as feedback inhibition. But we also have enzyme inhibitors that are called poisons, okay? So poisons such as cyanide can block enzymatic activity in the electron transport chain. And what that does is it stops us from producing, so this is my Xing out, stop, producing ATP. So we can't, and so maybe I'll do it this way, we can't produce ATP. And ATP, as you remember, is the energy currency of our cells. Penicillin blocks the active site on bacterial enzymes used to make a substance known as peptidoglycan. And when they can't produce this, since it's part of their cell wall, they no longer are able to produce a strong cell wall, and now they're more likely to be killed by the osmotic environment that they're living in. And then malathion is a inhibitor that binds to enzymes used in the breakdown of acetylcholine. So acetylcholine, for those of you who don't know, and probably most of you don't yet, acetylcholine is what binds to skeletal muscle or to our muscles and allows us to move. So when acetylcholine binds, then our muscles respond and we can move. So if this inhibits the breakdown of acetylcholine, the muscles will constantly fire and you will end up, or these insects end up in a tetany. And so this could also happen, since insects are animals, this could happen to humans too. If we had this malathion in our system, it could actually stop our muscles from being able to relax. All right, I am going to stop here. Oh, nope, I'm not. I'm going to do this last slide. I'm sorry, this is a longer video. After cofactors, then I will stop, okay? And so some enzymes actually use helpers. The helpers are either going to be vitamins, or which are typically called coenzymes, or inorganic molecules, metals, that are known as cofactors, okay? And so both are helpers to an enzyme activity. So if this again is my enzyme, right? And the molecule I'm putting in here is kind of shaped like this. So it fits, but there's a small piece that wouldn't fit. A coenzyme might fit right here, allowing this to bind and fit in perfectly and be all snug, like a bug in a rug, okay? So coenzymes help the enzyme do its job. All right, so I will stop there, and we'll get into the next video talking about oxidation and reduction reactions, okay? Bye.